So in a ovule, what we find is that the embryo sac that is there at the center and the egg cell within the egg apparatus of that same embryo sac is the one which gets fertilized by the form by the pollen tube that enters through the synergies down below, and this zygote that is formed due to the particular process of the fertilization will actually lead to formation of the embryo. Now this embryo formation at the same time it actually triggers the development or transformation of this entire embryo sac into the seed which also sorry, which also makes up or form its inside structure with the help of this entire ovule that is there, especially these integuments that are there in the surrounding. So they are actually the one that entirely transform into the seed. Now in some cases it has been noticed that some alternative things it does happen, alternative things other than this normal process of sexual reproduction. We find that instead of developing of one single embryo, sometimes there are development of two embryo instead. Two embryo they emerge out from the same particular place. If it happens so that this ovule it actually or this embryo sac it actually develop another set of embryo from it, maybe from this one or maybe from any other source. If there are multiple embryo coming out from a single embryo sac, that situation is what is known as a polyembryony. Polyembryony. Okay, means a single embryo sac is giving you, or actually multiple embryos can be obtained from a single ovule. That is how we can say. Okay, multiple embryo coming out from a single ovule. Now if I want to explain it even further, let me talk about a completely different step. Now this is actually by the process of fertilization that we normally get it, but there are also formation of seeds where fertilization does not takes place, a condition which is known as apomixis. Apomixis is a condition where seed develops without fertilization. Okay, without any fertilization. But not only that, the main thing is that the development of the seed takes place without even any meiotic division taking place. Because apomixis is different in that particular way is because that apomictic seeds they are formed without even any meiosis taking place there. So what actually happened is that inside the ovule that we normally find, if I just simply draw the ovule, we find that this megaspore or this actual embryo sac they develop after meiotic division of this nucellus, this area which is known as the nucellus. That from that particular nucellus the megaspore mother cell is formed, and from the megaspore mother cell this by meiotic division the embryo is actually it forms. All right. But in this particular case of apomixis, we are going to find that the nucellus is dividing directly into the embryo sac. It can form an embryo sac. There is there. Okay, that it can transform into an embryo sac. But that embryo sac will not undergo any fertilization of the egg. Instead, that the central cell that is there, both the pollen tube will enter here and will go straight away into this central cell and will form a primary endosperm from there. It's not going to form a it is going to form endosperm from there definitely. Okay, by different steps of division taking place there, but there is no fertilization of any embryo taking place in this area. I mean fertilization of the egg taking place in this area. Why? Because the egg is already in the diploid state. In the normal ovule, as we know that the egg is in a haploid state, in the case of apomixis, since there is mitotic division which led to the formation of the ovule or this embryo sac, is actually going to have a diploid egg right there, and that diploid egg will directly form, means here the diploid egg will directly transform into the zygote, and the zygote will slowly develop into the means embryo, right? So here we find that they are present in the form of like a diploid embryo, whereas here in this case we find that the polar cells are also diploid, but then two haploid cells means two haploid germ cells are going to enter straight away right here. Let me enlarge the view, then you will understand what is happening. So we have the pollen tube coming as normal as it is, all right, but the pollen tube which was supposed to fertilize the egg here, instead of going to the egg, it will actually go straight and go fertilize this polar cell. The diploid polar cell that was already there, polar nuclei, will now have two haploid sperm cells coming there. So ultimately a triploid nucleus is definitely formed again. Triploid endosperm is again formed here. Okay, it is 2n, both of them, whereas this is n, both of them are n. All right, so we are going to find that this is going to transform into like endosperm as normal, whereas this egg cell which is now diploid, this is a diploid type of egg cell, is going to develop directly into the embryo. It is going to transform into the embryo. So now what we'll get a seed from here. The seed that is going to be formed from this seed is going to be simply the like a normal seed that we normally know about, but this seed is produced asexually. It is not via sexual reproduction. It is completely asexual in nature. This actually helps in preserving desired quality, because by sexual reproduction there is a chance that a quality can get lost, because two different features are mixing together. There is a chance that the plant was having a very good quality in it, now that quality may be lost. But here in this case the quality will retain itself, because it is just the copy of the plant itself. Secondly, there is no involvement of any pollination, so there is no excess wastage of energy or material by the plant, so it is directly taking part in the process of formation of all these things, different things. Means instead of going for specific structure formation or anything, it is directly making the seed out of its own, right? So it is completely facultative growth all by the plant itself. So this method of seed formation, this method of seed development is what is going to be like somewhat many of the plant they find it quite useful, isn't it? So this is something that the plant they do this, especially like many of the plants. For example, we can set the example of opuntia. You know, this is a cactus. Okay, then we have the citrus plant. All of these plants, okay, they actually they follow this particular method. Then if you know this plant, I don't know whether you're familiar with this or not, it's called parthenium. Is actually kind of a weed that grows a lot in the shrub. Is a shrub type of plant, but they grow a lot. It's quite opportunistic and quite like vigorously they grow a lot, and it is considered because it produces the like some toxins in the seeds and all, and also highly potent allergen also. So this actually they grow a lot in our surrounding. They are brought unfortunately they are brought, this particular plant was brought by the accidentally they were being brought by the Britishers while they were exporting from their country, means it's importing in our case, this rapeseed oil, the rapeseed materials and all. So at that particular time, this parthenium, they got into the mixture and they came here, and then once they started growing in the field, as the parthenium go by this particular apomictic way method, so they produces a large number of the seeds from its own body, and they spread the entire area. They occupy entire space. You will find this plant, if you are familiar with the type, you will find this plant growing anywhere near your home, wherever there is a little bit of uncontrolled foliages or uncontrolled growth of plants taking place. In those areas you will find this particular weed growing there a lot. It's a kind of a weed. Okay, so this parthenium, opuntia, citrus, all of them, they go by this apomictic type of method of development. Now it is not only nucellus, but also the integument can also lead to the growth of the apomictic seeds. Okay, means not only the nucellus area is capable of doing this, but the cells of the nucellus can also develop into this particular type of like it can produce this apomictic seeds. Okay, so apomixis can be both from the nucellus region and also come from the this integument region. Keeping that in mind, if we consider instead of going for apomixis, if it is so that the embryo itself develops from this area, embryo develops from this integument or from the nucellus, or from like along with this like this area also, the normal seeds also, this kind of growth is what is known as polyembryony, because polyembryony ultimately means that from a single ovule, if you are getting multiple seeds coming out of it, multiple embryo mostly developing from there, so that considered as a polyembryony. And weirdly this these two groups of plants also undergo polyembryonic growth. Okay, and there it has been noticed that in it can form apomictic seeds from the integument and the nucellus. Also it can lead to formation of polyembryony from the integument and the nucellus. So these two methods are actually some additional things that I left out unfortunately, and is now discussed here in this particular video.